133 dichloro. Oh, the top one is cyclohexene.
I don't see any cyclohexanes.
What's
So that means benzene.
so it wouldn't be cyclohexene?
No,
It would be benzene?
it would be a benzene. But our parent molecule is going to be benzene for that one.
When can we switch that? Just that.
When we first learned, when you have three alternating bonds in a hexagon, that's benzene, because we get that aromatic character. A hexagon without the double bonds, that's cyclohexane. One double bond, that's cyclohexene.
Oh, okay.
Three double bonds, though, it's aromatic and it's its own thing. We haven't played around that much in recent, so it's understandable that you don't remember that. So our parent molecule is going to be benzene. And it's got a branch that's one carbon with an oxygen. So methoxybenzene, which actually does have its own common name. I believe that's anisole is its common name, but like I said, I'm not even sure I'm remembering that correctly. So methoxybenzene is fine. When in doubt, go the IUPAC name. Don't try to guess the common name.
Yeah.
You can always look that up if you need to. So for this one, cyclopentane, and it is going to be dichloro. Which one we call carbon 1? I would probably tend to put the ether on carbon 1.
That's what we did.
So that's going to be ethoxy.
Do you have to put a 1 in front of it?
Wouldn't be a bad idea, especially since we kind of hedged on which one should be number 1 anyway. Better to be overly certain. And then it's going to be 2,2 dichloro.
Why 2? Okay, I thought it was 2 as well, but
Sorry.
Epoxy is on
No,
mine.
you're right. You're right. It's 3. I counted wrong. 1, 2, 3.
Because I skipped.
Good
1.
catch, Taylor.
I went 1, 2.
Right, you did the same thing I did. You started. Yeah.
I was like, it's shorter. Okay.
And then we just have to do our resets. So highest priority is going to be there. Lowest priority is the hydrogen in the back. Then 2 and 3. So we get to the chlorines faster going down first. So 1, 2, 3. 4 is already into the board. So it's R. I have a box of Kleenex in my office.
It's fine. I'll survive.
The parent in me is going to go get that much to get another paper towel. All right, so good review here, especially reminding ourselves what benzene is, and that Riley and I need to work on our calculation. So really, that does make it a lot. This one we can give the old school name if we wanted to. I wouldn't even try giving this the old school name with the because it would be like 3,3-dichlorocyclopentyl ethyl ether.
Yeah. It rhymes better.
I mean, probably one of the reasons that the old names were the way they were is because you could speak them more easily. These are a lot easier to write and less ambiguous, but they're harder. They don't roll off the tongue the same way. This one we could do though for sure. When benzene is a branch, it's not benzyl. It gets its own name, branch name. So it would be phenyl. Not to be confused with phenol. Like phenol is with an OH. Phenyl is a benzene ring, it's a branch. So we name it as separate words, not run together and not with dashes. Methyl phenyl ether would be the old way to name this. That's worth reviewing mostly. When we get into esters especially, using the spaces instead of dashes or running it all together is how you indicate what's on either side. And that's kind of the same way here. And I also wanted to reiterate what phenyl means. This is not IUPAC. I'm not going to test you on this. If you said this to a chemist, they would know exactly what you're talking about. But IUPAC is the more standardized, and IUPAC, you would need to check the MCAT like study material to see if common names are part of it. I don't know that. My intuition is that they'll go all IUPAC because then it doesn't matter as much. But I wouldn't guarantee, I wouldn't put money on it.
Yeah. There's stuff
In the medical field in particular is really bad.
In dental, we had to do a whole like actual physical X-ray every year to just to prove that we could do it in case we needed to. Like we had to take it with the like little photos inside the mouth and then put them in the little photo machine and run them through all the three. And I was like,
it's all digital now.
Yeah, she was like, do this, but I have to make sure you can. We have a few physical prints on like that weird paper, and you know what we use for? When we have ear hematomas that won't stay up, buttons as a support against the back of the dog's ear.
There you go. Okay.
Or we did have to make a cone for a lizard.
A cone for a lizard.
It's not. That's so funny.
And you never, and realistically, a lot of like the spectra that we interpret in this class is a little bit of holdover, especially the mass spec piece. Like people still that work in the field still need to know how to interpret an NMR by hand because you still do that enough. But mass spec, you just feed it into a database and it gives you the answer back for the most part.
Yeah. With the extra small, and I asked them two of our big cones together. It was fat, hold on. But it was too long, but it's neck to it was too chubby. I could wear it as this kind of look you because it was two of our like 50s put together. Wow, that's the biggest one.
Dogs are funny. All right. This is not something that'll show up as a nomenclature thing, but it's more of just a term that I want you to be aware of. And it is actually showing up in medicinal chemistry and developing new, it's showing up a lot in drug delivery. Is this idea of a crown. They call it a crown ether because it literally kind of looks like an old school crown, kind of circular and kind of goes up and down. The important part is that we have these oxygens because these oxygens all have lone pairs. And so if you space them correctly, which typically it's every 2 carbons and an oxygen, 2 carbons and an oxygen. If you do that, you wind up with all these lone pairs pointed towards the middle. And it's kind of like a hemoglobin. You remember this? I don't know if you remember the structure of hemoglobin off the top of your head, but it was, and actually I don't think I could draw it exactly right, but it was basically like you get all of these 5-sided rings and they have the nitrogens have these lone pairs pointed towards the middle, and then there's 2 more at the bottom, so that you have another. And these are all linked together. The exact linkage is what I'm not sure about. And this works and is really important in biology because that pocket of electron density holds the metal in place, right? In your hemoglobin, there's an iron that sits right there.
Okay.
And that's what actually bonds to the oxygen, carries oxygen throughout your body. And so they took that idea and they applied it to simpler synthetic molecules, where we just, instead of having nitrogen, these heterocycles that are a lot harder to make, they just made a cyclic ether with a bunch of oxygens pointed towards the middle. And it does the same thing. You wind up this, so this is in this case, this is a they call this 18 crown E. It's 18 because it's a total of 18 pieces in the ring, and 6 of them are oxygens. And what that does is that's just the right size to fit a potassium ion, and basically hold the potassium ion in place. And that makes it really convenient as far as drug delivery, because basically you can leave it, it'll stay in this form, which is less soluble in water, but soluble enough in water. But basically the potassium doesn't get dispersed through the entire body. It only gets dispersed when it gets to a part of the body that has potassium deficiency or has a better binding site than this crowning. So they're seeing this. We're seeing this for they use this for platinum for chemotherapy, because platinum compounds are relatively toxic, and so they had it sort of designed so that until it gets to a cancerous cell, the platinum stays contained.
That's actually really cool.
It is. So this is one of the big overlaps. And it's not. I don't think they usually would call it pharmaceutical chemistry, because we're not just dosing that ion specifically. It's more about how do we get it delivered properly. So for whatever reason, I'm not actually sure entirely what the distinction is between medicinal chemistry and pharmaceutical chemistry. But I believe this fits more into the medicinal chemistry field. I think it's called, there's a similar idea with a molecule. I think it's called cisplatin. Yeah. So this one's not caged. Cisplatin is a chemotherapy drug. And it's just platinum with 2 chlorines, 2 nitrogens attached. And so, but it has, like, as you're aware, chemotherapy has lots of negative effects, right? Yeah. And because you're essentially poisoning your body, the trick is to get it to poison the cancerous cells without poisoning the rest of the cells. And that's where the drug delivery piece really comes in, targeting it and keeping it contained. So most of the reason we have no shortage of ways to kill human cells. There's plenty of chemicals we can use to just kill human cells, including cancer cells. It's doing it in a targeted manner and minimizing these side effects.
Like hit them until like a couple days after they get like is that like the body not responding to it yet?
It probably depends. It's going to depend a little bit on the individual's like immune system and metabolism and stuff. But I'm by no means, I've never taken an A&P class, so like this is just my guess. It probably depends also on what the exact chemotherapy drug is. A lot of them are basically just heavy metal poisoning. And so different heavy metals are going to take longer to accumulate in different parts of the body before you'll actually start seeing some of those effects. Educated guess, but still just a guess. The way we make so here's another example of a use for this. It also lets organic compounds be more soluble in non-polar solvents, because if you that same molecule from the previous page, we said, well, it's kind of soluble in water, but we basically took an ion, surrounded it with all of this less polar stuff. And so that's going to make it a lot easier to keep it contained and keep it soluble, which part of the drug delivery piece too, but also just in terms of synthesis. If we wanted to keep, like potassium fluoride is not very soluble in benzene. And if it's not soluble in benzene, it's not a very good nucleophile, because that's one of the reasons why we never really used fluoride as a nucleophile, is it was too stabilized in water. You want to see what the camera looks like?
You're talking about this here, excuse me, sorry.
Yeah,
This here was something.
I mean, it is, but.
it looks like that.
Oh, okay, it's got a yellow place to it.
And the resolution is lower than it usually is too.
Check out. Can you get to what app are you doing this through? Like
Let's do Zoom. I just use Zoom and record to the cloud to record stuff.
okay, what if you look in Zoom real quick? Go
Video.
yeah. Let's see what we got here.
You can drive.
Let's see anything standing out here.
Yeah, I mean,
Okay, let me go. Sorry. Yeah.
no worries. We'll be here. All right, so just as a recap, and again, this is not something that will really show up much in the class. I want to be, this is just a good reminder of fluoride is not a good nucleophile in a protic solvent, because those protic solvents stabilize it too much, but it also won't dissolve in a nonpolar solvent. So crown ethers are a good way to get the fluoride to dissolve by basically stabilizing half of it really easily. And we can kind of tailor it to whatever we have, whatever the bigger ion or the cation is, we can change the size of this to fit it.
Okay. Bigger meaning like actually in size or like?
Yeah. Yeah. So like potassium's got a different size than sodium, right?
Yeah.
Sodium would be smaller than this, so we probably use, instead of using 18 crown 6, we might use 15 crown 5. Again, be aware of it conceptually. It's not really going to show up as like a practical thing we're doing in this class. But just the idea of a crown ether and the idea that we could stabilize, we could use these lone pairs to stabilize things. It came from biochemistry, so it's a concept that'll come back when you take your upper division biochemistry after your transfer, which you should be looking forward to. That's a really hard class, but it was one of my favorite classes. We spent like a whole month just on hemoglobin and the structure of hemoglobin and we were talking about the entire protein, rather than, that's called the heme group. It's called a prosthetic group that is attached in the middle of this much larger thing. And also, it was 20 years ago. Okay, give me a break. It looks pretty similar to what I've saw, as long as we're looking things up. A porphyrin ring. I can't see what I'm typing, so I think I missed it pretty badly.
Porphyrin does not exist. Oh, you have a double piece. Yeah, 2 pieces.
I was close.
Yeah, I'm trying to look down here. You want me to just
Hang on one
Yeah, you're
second.
good.
Let me just pull this Wikipedia page up. Am I in the right spot? I'm at an angle.
Yeah.
That was pretty close. All right, go ahead.
sorry to interrupt, but I think this might be a quick fix.
And in different things, so basically this whole thing is basically sits in a pocket in hemoglobin, the entire molecule. And the hemoglobin is made up of 4 different polypeptides. So hemoglobin the protein in the red blood cell that carries the oxygen.
But isn't it what makes her look like if we're bleeding like it falls up?
No, I think that's actually called globule.
Oh, cool. Okay.
hemoglobin is, it's hemoglobin is we spend so much time on it because it's really good, it's really well understood protein, but it also is very complicated and very subtle in a lot of ways. So it's actually got 4 different polypeptides that are all linked together. And each polypeptide has one of these that sits in like a specific pocket where there's like a covalent bond that comes off of some of these corners, holds it in place. And then with that, the iron sits right here in the middle. And the iron being positively charged is a place for oxygen's lone pairs to donate. And so the oxygen molecule comes in, and because it's sp3, it comes in here and it sits at like an sp3 angle because the electrons from the oxygen are at a trigonal planar angle. Exactly. But we don't want it to bind too well, because if the oxygen binds too well, it seems like, oh, high binding affinity for oxygen, that'd be a good thing for our blood to have. But our blood also needs to know when to let go of it.
Right.
And then basically hands it off to a molecule called myoglobin, which is present in the muscle cells. Did it look like did that work?
Man, y'all are deep in conversation. I hate to interrupt. This is terrible.
Y'all are on a roll. I hate when Zoom defaults to the
I'm not even seeing the camera now, is it?
yeah. Hang on. Oh, I moved it over there. That's why I'm not finding the important one. These dual screens, when they're behind you.
Not super great.
All right. Now let's see if I can turn it back on. That looks good.
Is that better?
Yes. Yeah, the color's all good. Nice work.
Excuse me, thank you.
Thank you.
I just have to get it focused back in on us. Wonder if I can set these presets so I don't have to do this by hand every time. Should look into that. All right. We're back.
Is it better?
I think so. Yeah, you can't see what I'm seeing, can you? Yeah, color's good.
Yeah.
All right. So let's look at some other reactions of ethers. When we look at how we make ethers, the number one way we see it is we're just going to do an SN2 where our nucleophile is alcohol. So we saw this before. We've made ethers before. Well, not in lab, but we've seen this reaction before. So you can actually start with a primary alcohol and then use an acid catalyzed reaction to make the alcohol that are leaving group, and then have your alcohol come in here, do to attack, and then do a proton transfer to remove it. That gets you okay yields. The problem with this is this really only works. One, it only works for primary alcohols. And 2, it only can make symmetrical ethers. If you try to do this with a different alcohol, you'd wind up with a whole bunch of different products. You'd wind up, if we tried to, instead of this being ethanol, if we made this isopropanol, we could make this a different ether, but then we'd also get the diisopropyl ether. And then we'd also get still get the diethyl ether, we'd get the one we actually want, but then we'd also get a third one that would look like this, because we're really just going about it in terms of like statistics and probability. So this is the simplest way to make an ether, but it's not very useful unless you're trying to make a symmetrical ether that happens to be a primary alcohol. Starting from primary. So we couldn't even use isopropyl alcohol to example.
So what we use instead is called the Williamson ether synthesis. And it actually is pretty straightforward. It's reactions we've seen before.
Did the one before have a name?
Just acid catalyzed dehydration of primary alcohols. But because it's not all that useful, it doesn't get its own name. The Williamson ether synthesis is a similar process in that it's still just going to be an SN2, except we're just going to start with an alcohol, and then we're going to deprotonate the alcohol to make it an even better nucleophile, and then we're going to add an alkyl halide that has a good leaving group. Now that I've gotten used to having a clean board, I don't think that's even honest.
Yeah.
Did one to deprotonate this out of low?
So one deprotonates, and two, we add an alkyl halide that has a good leaving group on it, it's like a chloride or a bromide. And we just do an SN2 with it. So if you think back to when we first learned SN1 and SN2 with the bromides and the chlorides, we frequently had like sodium ethoxide was oftentimes one of our nucleophiles, right? Step one is you just take the alcohol and turn it into the deprotonated form. And then you do an SN2, just like we've been doing since October. Right, so really the Williamson ether synthesis is nothing we haven't seen before. It's just framed in a different way because like, oh, if you want to make a specific ether, here's how you do it. And so here's our mechanism here. Sodium hydride. Remember, we saw this before when we first started talking about alcohols. This is just a way to irreversibly deprotonate the alcohol, so it's not a reversible step. Because when we do this, the hydride comes in, pulls the hydrogen off the alcohol, it makes hydrogen gas, and it just leaves the system. So it can't happen backwards.
Yeah.
Right, we could do this with just any strong base like sodium amide or something like that, but that's always going to have its own side effects. So this is just a really convenient way to deprotonate it and keep it deprotonated. And then once we do that, we just do an SN2. Basically, our only limitation here is we can't do this SN2 step on a tertiary halide.
Okay.
Because if it's tertiary, it won't go through SN2, right? Just because of the sterics.
Yeah.
But what we can do is switch the red and blue.
Right.
Because this first step is not limited by sterics. So we can do this first step no matter what part is, whether it's primary, secondary, or tertiary. We just can't do the second step if the red one's tertiary.
Okay.
And we do have to worry a little bit about, we could make this into a sterically hindered base. So there's going to be some side reactions, some elimination products that might happen.
Okay.
So we might need to make sure we're doing this at a cold temperature to eliminate the elimination side effects, especially if the blue one is tertiary.
Okay.
So there's some practical things to consider, but it's still a pretty useful reaction. You ever get a piece of pine needle stuck in your sock?
Yeah.
Like you can't get it out, but you do, it just sits there. Yeah. Alright, so how does this work out? We're trying to use Williamson ether synthesis to make MTBE, that gasoline additive. We have to choose our steps properly. The first step works no matter which way we do it, but if we're trying to make a tertiary ether on one side and it's a methyl ether on the other side, we have to start by making the methyl ether, or sorry, the methyl alcohol into our nucleophile. Sorry, we can't do that because it can't get in there to attack this tertiary carbon. We need to deprotonate the tertiary alcohol and turn it into a good nucleophile and use the methyl is our target for it. Right, because tertiary carbons can't go through SN2. But the nice thing about this is a little bit like the Grignard reaction. Remember when we were like, oh, we can do this in 3 different ways. We can make this depending on what order we do things and where our bromide is. It's a little bit like this. There's always 2 ways Williamson synthesis. There's always 2 options. One of your R groups starts as the alcohol, the other one starts as the halide. And you can always switch them if needed. So let's do a practice.
Okay.
This one you can do 2 ways.
You want us to do both ways?
Let's write out both ways and let's see if one of them makes more sense than the other. While you do that, I'm gonna go sit down in my office and try and find this, whatever's sticking my name.
Okay. Okay, so. Do you want to drop this for each thing? You do it, okay? And those, yes. Okay, so first. First thing we're gonna do is. And we're gonna have NaH. And the Na. Positive. And the H has one. And the lone pair. We getting to that or starting? Oh, my problem for me. And we're starting. That's the goal. I think you're right, because it's showing me it's like there's a lot of feet. We may keep following. So if we started with the everything, yeah, and we have the oxygen, and let the oxygen. Sometimes maybe we say a little bit, you know, they gave rules as they go. Is that our goal? Is that what we're?
That's our goal, how do we make this ether?
So then we have our positive H. It's lone pairs. Oops, hydrogen is one function. And the bond goes to the oxygen. Oh my God, you're driving up with rain on the nose, girl. Keep going and I'll keep talking about that. Yes. I am with you. Look, so now we have a ring. And that ring has. Entanglement is. Eight, seven, eight mates. Okay, so I have 6. And then we still have our ending positive. And now we bring in our next group. Okay, so I'm gonna use this one. I'm gonna use it. Okay, you with me? This is our RX step 6. That's RX, these are both substitute for technical values. So I'm using a single X. And X is methane. Well, but it could be chlorine, it could be bromine, it could be chlorine, it could be connected down layers. I'm going to tell you. It's always been followed. We need to talk. Well, I said it. And then one of these lone bears is going to go to our arbiter. So would you just follow along, please? And then the dog is going to go to the X. Okay. And so then that gives us. Oh. We need more. We need more R. Yeah,
So maybe not methyl,
we want to use an ethyl.
there you go. But your mechanism looks like it's drawn properly.
And I'm catching my mistakes, which is good.
Yeah,
Here, instead of RX, it's like RRX. If that makes sense. And then we have our energy. So. Okay. Now, the lone pair, that's where, from the lone pair from the oxygen is going to attack this case. I think the same carbon still, because then we still want that one to go to the next phase. Now, have a ring. We have our ring. And we have our oxygen off of it. Now that oxygen has the ethyl group. Yeah. You feel me? And then plus, yeah, and then with this. Boom, that's one.
but now you've done it once, the next one should be easy, right?
Yeah, I think the next one makes more sense. We were drawing out the full mechanisms, make sure we got it.
Yeah, that's what you should be doing right now, it's perfect, it's good practice. Since you did it properly, I'm not going to do it up here.
Yeah. It's kind of. Users confirm.
So since it does ask for specific reagents, I just want to pick one. Iodide or bromide are really common for the small R groups because they make it so that it's solid or liquid at room temperature. If you try to do ethyl chloride, that's going to be a gas at room temperature, and we don't want to do a gas. So once you have one of them, all you do is you switch what's the alcohol and what's the bromide. So our other option here would be ethanol plus NaH makes the ethoxide ion. We're just following our normal pathway that gets us to our final product. Does anything look weird though?
For the second one, don't we want to use iodine instead of, or does it not?
Doesn't really matter for this one because it's on the bromine. Bromobenzene is the liquid. You can even use chlorobenzene if you want to, except for the fact we've never seen an SN2 reaction on a benzene ring before. Is that going to have a partial positive that's a good target for a nucleophile? There's a whole bunch of electron density here.
Yeah.
So basically, benzenes are not going to be good targets for SN2 for nucleophiles. So that answers our question about which way makes more sense. The first one makes more sense because we know we can do this. We can deprotonate an alcohol here. In fact, phenols are more acidic than most alcohols. So that's even easier than normal. And turn it into a good nucleophile.
Because
For
the
red.
last, can't, right?
Because this last step can't happen. Basically, we're not going to see SN2 reactions on this carbon is an sp2, right? Because trigonal planar it has a pi bond. A carbon that has a pi bond is not a good target for a nucleophilic attack. It's a good target for an electrophilic attack because it has lots of electrons, but that's not what we're doing.
But we usually like it or stay in those places. If it were only like a tertiary, the trinary, we would move like the positive. Like,
On this carbon,
to make it more stable. If it wasn't 4 months, if it was 3. Right?
I think I see what you're saying. Yeah.
So like it doesn't, that's how I'm like making sense of it. It's like usually we would put a partial positive or partial negative there to make it more stable so that we like, don't attack it. So,
Right, so it looks like this carbon should have a partial positive because it's attached to a bromine, but because it's got all these other electrons, and even when we got the benzene ring factor, we never saw this react in an SN2.
right.
Right, if we tried to attack this with an SN2, usually what happens instead is we wind up breaking the pi bond. We can't break the pi bond on a benzene ring because that breaks up the aromaticity. We won't ever see well, we have a whole chapter on that coming up. But basically, if your target looks weird, if it's not just a regular alkane, that's probably not something we want to do. I can't say that we never break the aromaticity, but we wind up with something called electrophilic aromatic substitution when we get into our chapter on aromatics. That's not nucleophilic. Right, and then one of the other nice things about ethers is we can undo that too. The same way that all of our substitution reactions basically, if we change the conditions, we could get the substitution to go either direction.
We undo what this.
If we start from an ether, we can actually break it apart back into its pieces, into its arbiters. Yeah. And it actually takes it a step further
we have to do this with excess acid, and has to be a hydrobromic or hydrochloric or hydroiodic. So HX is the placeholder for that. If we use a whole bunch of strong acid heat, we basically just do a couple of substitution reactions, and we wind up breaking the ether apart. So for instance, if we had 2-ethoxypropane and we exposed it to excess HI, and heat, what's going to happen is that first off, you're going to protonate this, which makes it a better leaving group, and then your iodine can come in here, and we just do another SN2, just the other direction. Instead of your halogen leaves, this is the mechanism. Instead of the halogen leaves to make room for the carbon-oxygen bond, the carbon-oxygen bond leaves to make room for the halogen. So that turns it to being, now we've got isopropanol and ethyl iodide. But if we have enough of this HI, we're adding heat. This one? Yes.
Why? He's color. Okay.
Like we have up here.
Oh, okay.
I'm showing you that we have 2 different colors. It's like, yeah, I guess
It's trying to be.
it's this color.
It's brick red.
Yes, there you go. It's brick red.
I just want to make sure I'm colored.
Yeah.
Yes.
So, and I could have chosen either of these to start with. I didn't, there's nothing special about the ethyl group versus the isopropyl group. I just picked the one that was closer to the arrow. Because then the next step is, if we still have more HI around, it does that again. We can still do a proton transfer step and protonate. So then now we have an even better leaving group, and we still have iodide around. So our final product is still going to be, we have the ethyl iodide, the ethyl that was part of our ether turns into an iodide, and then the isopropyl group that was part of our ether also turns into an iodide. This is why we spent so much time on SN2 reactions back in October, because everything is built around them.
So you have 2 OHs going there?
So then we wind up making water as our byproduct. 1 water. Because there was only 1 oxygen at the beginning. All right, so they call this acidic cleavage. Cleavage just means you cut something in half. Okay. If you want, I have the neater version on the next slide.
What is it?
I was trying to say, show that this was not, I didn't want to use the brick red to confuse it. So you could just use the blue too. Because
It's just different.
the carbons are the same blue carbons. The iodine just comes in and pushes the OH off, the H2O.
And then we have just
Plus H2O. Why is the heat helpful in this situation? What is the heat doing? I mean, everything's more likely to go, or everything goes faster when you have heat. What is heat vapor?
entropy.
We started with one object here and made three, broke it up into a bunch of different pieces. So the heat favors this side. We don't necessarily need the Williamson ether since this doesn't require heat because we're trying to make something into one larger object. We don't usually want high temperatures for that. Well, it sounds like it's a good time to take a break then. Right, so proton transfer, nucleophilic attack to make the alcohol and the halide. Then I'm just reiterating what we already did, just showing you on either methods. We are taking our break. Let's come back at 5 after. We'll go over this one more time after we've had a break to solidify it in our brains. And then we'll move on to epoxides. We'll practice first and then epoxides.
I want because you're looking at it. So it's small.
All right.
Bye. Have a good night. Always a good day when we keep that. Always a good day when we keep that. Perfect. Okay. Every time I say that, he does.
Sorry. I got pulled into a hallway meeting. All right.
Maybe we should go search.
No, we're making good time today, so I think we're okay. Or else I would have cut that off earlier. All right.
Should have just left this one up. We could have been working on yours.
I almost thought about hitting the next, but I just got what was there.
You always can.
That's totally fine.
This is where to get back. But it's good because. Do you want to?
Just the products. You can show some of the steps or intermediates as necessary.
This side, this side.
You got the water. Yeah.
Well, that's it.
Per day,
It's gonna be the same,
there's manageable.
but
So you
I
can
still
see.
show that I had the two sides, so.
And if you wanted to show it, draw it twice to show the, how it breaks apart, that's fine too. This would be more commonly correct though.
But B, isn't it gonna do the same thing?
These all do the same thing,
Oh, we do.
like the reactants for all of them.
Okay.
It's just practice with ring opening reactions. What are the pieces turn out to be? Can we do that if it's benzene ring? Probably not, so we're gonna not wind up with two bromides, we'll get phenol. And then this one will turn into a bromide.
For B, we open the ring and is it on each end?
We're gonna break both of those off, to make our water molecule, right? So 1, 2, 3, 4 carbons, they're all attached. I have that here. I have that here.
Plus H2O.
Plus H2O. So for C, like we talked about, we can't do the SN2 on a benzene ring, so the oxygen stays attached to it. But the other side gets turned into a bromide.
But something like that, if we were doing, so isn't there H2O stuff? Yes.
No,
Oh, no.
because the oxygen stays attached to it.
It if we were gonna do, let's consistent, one of those parts, this one would be a good way to do it, right? Because of the wheels.
I mean, if we had a bunch of this for some reason, like a lot of times in synthesis, actually commercially. Like you wind up finding somebody else's waste product that has a compound you work with.
Okay. So if we had a whole bunch of it.
If we had a whole bunch of it, this is not a bad way to do it. There's faster ways to make phenol, but if you had a surplus, why not?
It's- This part of this, it would just flat, and then this one, we can't. Hold it on 2 times, right, so it's a little bit.
But back in the redraw. I was not done with it, I was gonna add the methyl group, and then I decided that I was gonna redraw it with everything in the same spot, so I could keep the stereochemistry, make sure, because it's an SN2. We're not worried about the stereochemistry for this carbon, because it's the end of the carbon chain, but this carbon is gonna have a specific stereoisomer. Because it has a methyl, it's gonna have an iodine, it's gonna have this whole thing, and then it's gonna have a propyl group, so it's got 4 unique things attached to it. So the way that I would approach this, as soon as I realized that, I said, okay, well. If I need to make sure I'm just going to save this and then flip it when the iodine attaches. I'm gonna redraw everything in the same spot.
Both of it.
I'm gonna put the iodine over here. The oxygen is here, I'm not gonna flip it yet. So when the oxygen is going to leave. The iodine is gonna come in from this side and these are gonna get pushed that way. And so I'm just going to. In the interest of showing this properly. So now my methyl's there. My propyl's over there. And my iodine is here. Right? The oxygen left and I just pushed those two that way. Which then we could clean up if we wanted to. If I was in a time situation on a test. And I'm worried about what, I don't want to clean it up. To the point where I risk messing up with stereochemistry that I was very careful about.
Yeah.
And I also don't want to take time. To necessarily go through the process of assigning R versus S and making sure it stays the same. On a test, I'm probably leaving it like that.
That's a full credit answer. I'm confident about the stereochemistry because all I did was that umbrella flip. So I'm not even gonna try to redraw it. If it's not a test though,
Well, they'll just understand, they didn't switch.
it did, it got pushed that way and the oxygen left and the iodine attached on the opposite side. So that's what flipped the stereochemistry.
Okay. I was thinking like forwards and back.
You could do that if you put the iodine right here and you just flip those two,
Yeah,
that would work as well.
that's what that was my.
I think I like to think about it this way because it reminds me that the iodine is coming from the opposite side and everything else moves away from it.
Okay.
It's a little bit closer to the actual physical process, which helps me visualize it. Then we'll end with another easy one. Got both of those, so it's cyclohex. It's bromocyclohexane. And these are not the same. It's a common mistake to make.
They're water.
Yes, plus water.
What about for?
Also plus water.
Oh, except C don't have one. Oh, except.
C and D do not have water because the oxygen that would make the water stays attached to it.
Oh my God.
And again, that only applies if it's directly attached to it. If there was one carbon in between, then it behaves just like a normal carbon. It's only because the oxygen is directly attached to the benzene. Yeah.
Oh.
I did start with that really good yesterday. He was walking around here, on campus. And I saw him through the corner window of the lab walking that way, and so I jumped out of him from the doorway, and he fell over. We knocked him down. Yes, he's better.
That makes it so much better. It's gonna heal in a week.
It's been 3.5 weeks since the first one. And the second one was just aggravating the first one.
I think Bruce did that the other day to someone.
It's kind of fun, when you're sitting at the front, you're watching people walk
Yeah.
by, then you get the timing down.
He did exactly. Them walking, it's like, and like scurrying that out.
Yeah,
But it was funny.
that corner window doesn't serve a whole lot of purpose.
Just look at. Yeah,
Do you?
yeah, I like class and then I went up and it was funny because literally like 20 minutes before, we had talked about how he's like very quiet on his feet, so he always startles his wife on accident.
All right, we're gonna come back to this for more practice if we have time. There is a square. It's like, well, it's really unstable. It's still just a carbon ring, it's just 4 carbons, maybe ring instead of 5 or 6.
Are you gonna do that on the exam, throw a square that you've never seen?
Maybe a
before?
heptagon. I do like making you draw a 7-sided ring.
Say, yeah, I think you did it on.
I did it on the final or maybe the midterm last term.
So A is O's and O's, so there A and B are.
Yeah.
And then another step after you have two products.
It's not gonna be 2 products
Oh,
because it's still attached.
okay.
But then what is this step?
Oh, it's one of those that just. Our yesterday, we did it our.
Yes.
No, it was last week, it was on the inside project quiz. Well, but we redid this one. Yeah. Breaks double bond.
Actually, you know what? I like this as a quiz question for the weekend. So the quiz was pretty light. Yeah, so I'm gonna leave this for now,
Okay,
and we're gonna skip this. And I'll put it on the quiz this weekend. Thursday.
I know, this week was really fast.
Tell me about it.
Like. Two. Okay, what's this? Epoxides.
Epoxides. Another ring opening run one. So if we want to make epoxides, there's some ways we can do that. We have two methods for it. But the only thing that's going to show up on the quiz this week is naming them. And basically, we just throw epoxy as a prefix.
Like, menthol epoxy.
More like 2,3 epoxy pentane.
Okay.
So it's for whatever reason, we don't specify both ends of a carbon-carbon pi bonds. Right, so we wouldn't say 2,3 pentene. But we do specify both sides of an epoxide.
Okay.
So anytime you see epoxy, it should have two numbers. It's this piece, yeah. But other than that, their naming is the same. It's pretty straightforward. So let's try it.
Does the epoxy, is this line part of the epoxy?
Yes.
But then it's not between two.
So find your longest continuous carbon chain. You're still gonna name it like it's an alkane.
I did, but it's not between two on the chain, that is just part of the epoxy. You don't have to count it.
It's really the oxygen. So the carbons can still count as part of the carbon chain. Thanks.
To
I don't like that color.
epoxy propane.
It's
Epoxy propane.
1,2 epoxy propane. You don't really need the 1,2 in this case. It has to be between 1 and 2, and we just come from the other side.
I was just naming it next to that.
And then there's a methyl group, right? So our parent molecule is propane. Take your pick. And again, this is a small enough molecule that really all of these numbers are redundant a little bit. But when we start getting these more complicated small molecules, it's not a bad idea to write in. You can't have epoxymethyl propane any other way than this,
Right.
but if we're just covering our bases.
Yeah.
Let's do C next,
Oh,
just because it's easier. We'll start with a simpler one before we tackle B.
big one.
Mm-hmm.
Oh, damn it, I forgot to get paper when I was upstairs. Okay, okay.
What are we gonna do for B?
Well, it's symmetrical, which is nice, because then whatever it is, you can just say, but it's.
What's our longest continuous carbon chain that has the epoxy curve?
The. That have the epoxy, then you count.
We never count these two over in, right? So our longest continuous carbon chain that has the epoxy curve. Just two. Now, we really don't need the numbers for the epoxy, right? Because there's really only two carbons. So it's gonna be epoxy ethane.
Two die cycle, die. Then
So the one.
what? No.
It's it's not phenol. Then. We change the emphasis to really make it clear. It's phenol puts the emphasis on the OL
Right,
versus phenol is when it's a branch.
Okay.
So it's gonna be diphenyl. And these ones do need numbers. I would say 1, 1 instead of 2, 2. But we do have to specify the numbers because they could be on the same carbon or they could be on opposite carbons. You could have 1, 2 diphenyl. It would be a phenyl group here and a phenyl group there.
that would be a what, a 2, 2?
That would be 1, 2. 2, 2 would be the same molecule we have. So they both be on the same carbon, but they could be on opposite carbons. So that's why we have to specify both of them. Look at all this good computing of our nomenclature today.
I like nomenclature, it's very just like puzzle piecey.
It's very cut and dry, and once you learn the process, it's the same every time.
Yeah.
There's little wrinkles, but that's it. All right. Couple more slides. We might even, we're not gonna finish early today. We'll finish today though.
Yay.
Epoxides, we have. I don't remember, this first one made a halohydrid, and then I think. Then we deprotonated. So we didn't spend much time on the bottom magnets, so don't worry about the magnets, but the bottom one, this top one, we've seen them before, right? Rocco, more recently than you would like on the midterm.
Yeah, unfortunately.
So. We're not gonna draw out the full mechanism here, but this is our go-to for making epoxides. For now. Next week, we actually get to cover a Nobel Prize winning reaction. And they won the Nobel Prize after the year 2000 for it.
Okay.
So we're getting current. He's still alive and still working.
I get to learn.
Yes. Not able.
People get.
For his epoxide formation, it was in 2001, and then he won a second Nobel Prize in like 2018. So he's one of very few people that has won 2 Nobel Prizes. And he's still alive. He works at Scripps Institute in San Diego.
People
It's affiliated with UCSD.
have won Nobel Prizes after they've died.
No,
Oh, they have.
that's one of the tricky parts about it. They don't award them posthumously. So posthumously means after you die. So that was the Nobel Prize organization's argument for why Rosalind Franklin didn't get a Nobel Prize for the discovery of DNA.
Yeah.
Was because she was dead before Watson and Crick.
earned it, but they didn't even mention her. And they only came back with that argument like in the last 10 years.
Right, after people were like, hey, actually.
Actually. All right, and because I want to spend time on that Nobel Prize winning reaction, because it does involve stereochemistry, basically what Sharpless did to win a Nobel Prize is he found a way to make a, starting from a flat sp2 molecule, he figured out a way to preferentially make one stereoisomer, which we've never seen before, which is why it won a Nobel Prize. Everything to that point was, well, it's the same top and bottom, it's gonna make full mixtures. And that is what we see for our other two reactions. Sharpless figured out a really complicated way to do it, to only make one of them.
So it's like Markovnikov versus anti-Markovnikov, but in this case, now it's sterics.
Now it's steroids. Now it's, yeah. So we're gonna just skip that for now though, and come back to it. So just so we can remember how ring opening reactions work for epoxides, because those are simpler. We've seen them before. Basically, it's just gonna be an SN2 mechanism and then the proton transfer. Any strong nucleophile will come in and attack the opposite side. You're gonna get an anti-addition, basically. So strong nucleophile comes from the opposite side. So it means that they're gonna wind up facing opposite directions. So because in this case, it's such a simple molecule, it doesn't work very well, but let's go back to that. Just the cyclohexane one. If we did this with this molecule, mechanism would look like that. And when it was done, we'd wind up with the trans product. Okay, so that's what I mean by anti-addition. It's similar to an addition to an alkene, and it's anti because we're winding up with things on opposite sides.
It'd be in that first step.
And it would be in that first step. The nucleophilic attack is where that happens. And then technically there's a second step. It's just the proton transfer because we have an unprotonated alcohol. And this does happen even faster than a regular ether. Like, so a regular ether, you can do this. We saw we could undo ether formation with relatively weak nucleophiles earlier on. Because it's a three-sided ring, those three-sided rings have a lot of strain energy. This reaction happens pretty quickly. And it basically means that any of our, if you think back to midterm of last quarter, actually the final of last quarter, we had that table of here's my strong nucleophiles.
Yeah.
Any of those strong nucleophiles are fair game. So a deprotonated alcohol, like we've already seen, cyanide molecule, a sulfur with a negative charge, a Grignard reagent. We didn't actually put that on our list before because we didn't know what a Grignard reagent was. Grignard reagents are strong nucleophiles. So this is actually making an epoxide is actually a really useful tool because then we can add more carbons to it if we want to. And same with lithium aluminum hydride. We can add a hydride to that other side if we wanted to.
I cannot spell Grignard to save my life. It's so weird.
Think of it like lasagna.
I don't know how to spell that either.
I don't know. I grew up with my mom making lasagna all the time, and my dad never failed to call it lasagna.
So every single time, once a week growing up, I heard my dad say Blazag. And that helps me remember, it's always the G then the N. It's basically the Italian equivalent of an N in Spanish.
I was trying to say like terme, like that is, yeah.
So in Spanish, you do this. In Italian, you put a G in front of it instead to get the same effect. So grignard, it's grignard.
What is the better word?
Nope. Look at that, we did finish early today.
Oh, that's it?
That's it. That's the last thing for today. We'll come back to epoxides next week and talk about sharpless in that stereo-specific epoxidation.
Yeah.
His second Nobel Prize winning reaction is one of the ones that I actually worked on in grad school.
Really?
I did the computational chemistry. I was trying to explain why it worked. It was recent enough and useful enough that everybody was using it in the synthetic chemistry space, biochemistry. But nobody had described it properly computationally, what the actual mechanisms and energies look like. So I spent a fair bit of time looking at that reaction. Yeah, and then you want to know the price for it. I never got to meet the guy. What's that? No, I didn't like my research, so it never got published. Which is why I don't have a PhD, is because I didn't get along with my advisor.
Really? That's